Hello everyone, welcome back to Biotech Hub. We are continuing with our research and methodology series.
Sampling techniques in research. Basically, the sampling techniques can refer to as the methods which are used to select a subset of individuals or a unit from a larger population for inclusion in a study. So, sampling is crucial for ensuring that the research findings and the representatives are generalizable to the population of interest. And there are mainly two categories of sampling: probability sampling and non probability sampling. So, let's dive deep into both these sampling types. So, probability sampling technique is a simple random sampling. So, when we are talking about a simple random sampling type of technique, then we can state that it involves randomly selecting the individuals or uh, or some units from the population where each member has an equal chance of being selected, and it requires a sampling frame that lists all the members of the population. Next, we have stratified random sampling. So, divides the population into homogeneous subgroups based on a certain character. So, in simple random sampling, we are selecting the individuals very randomly. Um and there was an equal chance of selecting any of the individual. But in stratified random sampling, we're dividing the entire population into some subgroups based on certain characters. So now if we'll select, then there won't be a equal chance of being selection. And then a simple random sample is taken from each of this to its size or importance. So next we have systemic sampling. So systemic sampling basically involves selecting each nth individual from a sample frame after randomly selecting a starting point. So say suppose we have 1, 2, 3 till a 50th number. So from we have started or we have selected number 14, say suppose. So it is involving selecting of every nth individual from the sampling frame after a random selection. So after 14, every nth number is being selected. And it provides a systemic way to select a representative sample, especially when a complete list of the population is available. Cluster sampling. So cluster sampling basically divides the entire population into some groups and then selects some of the groups randomly, and all the individuals within that selected groups are also included in that sample. We have an entire population of, say suppose, 10 individuals, and then we created three groups or three groups of three individuals each, or maybe one of a four individual. So now we have randomly selected this particular cluster or group. So now every individual which is present in this group B will get selected automatically. And often it is used in a difficult or a impractical situations to create a sampling frame. Now let's talk about the non-probability sampling techniques. First is convenience sampling. So convenience sampling involves selecting the individuals or the units that are conveniently available or easily accessible to researchers. And they are often used for its convenience and practicality, but they may result in a biased kind of samples. Second, we have purposive sampling. So purposive sampling involves selecting the individuals based on specific criteria or characters that are relevant to the research questions, and they are used when the researchers want to focus on a particular subgroup or when certain individuals have a unique insight or experience. Snowball sampling. Snowball sampling, it begins with the initial set of participants who are then asked to refer other potential participants, and it is used in a situation where the population of interest is difficult to reach or to identify. Lastly, we have quota sampling. It involves selecting the individuals to meet a predetermined quotas based on certain characters like age, gender, or occupation, anything, until the quotas are filled. And they are used to ensure that the sample reflects the proportions of different subgroups in a population. So each sampling technique has its own advantages and limitations, and the choice of sampling methods depend on the factors such as the research objective, the population characters, the availability of the resources, and the practical considerations. So researchers should carefully consider the strength and the weakness of each of the techniques to ensure that their sample approach is appropriate for their study. So thank you everyone for your patient ears, and please like, share and subscribe if it was a useful video for you, and stay connected with Biotech Hub. See you all for the next one.